All right, so as Dan and Christina said, my name is Aaron Sato. I'm the CSO of Twist Bioscience. I'm also the head of the Biopharma team within Twist. That'll become more apparent as I go through the presentation. Really excited to be back here at PEGS. It's been 3 years since I last spoke. At Twist, we know that the race is on to find the best novel biologic drug candidates, and we really understand that to take advantage of multiple technologies, multiple systems, help others discover, optimize, and develop drugs quickly. And so I hope you can appreciate by the end of this talk how Twist, and in particular Twist Biopharma, is bringing on all kinds of different technologies powered by DNA to help others discover as well as optimize antibodies. So Twist Biopharma, we're actually a vertical within Twist Bioscience. You're probably familiar with Twist as the company you go to buy lots of DNAs, and that's actually our primary core business. And as you'll see in a second, we use oligopools to generate all the custom DNA products that Twist sells. And those sit in our synthetic biology division of the company. We also have a lot of products and use oligopools for NGS enrichment, and that's also, again, a core DNA product that Twist sells. The two ways we kind of elevate those products is creating two extra divisions of the company. One is Biopharma, which I'll spend a ton of time talking about today. And then the second is actually data storage. We're actually storing data for long-term data storage in DNA itself, which I won't talk about today, but if you want to drop by and talk at the booth about DNA data storage, please feel free to do so. I'm a big fan of reading management and leadership books. One of the books that I love, it's my favorite book of all time, it's called The One Thing. And the reason why I love this book is because the premise around the whole book is that the best companies out there, the best leaders, the best people really understand the one thing they're really good at. And so for Twist, that's our ability to print DNA. And it actually rests upon this silicon chip shown here in the middle of this slide, where we can actually print up to 1 million individual oligos, up to 300 base pairs in length. So it's an amazing printer for oligopools. And you'll see in a second, we actually use those oligopools to make all kinds of really cool custom DNA products. In particular, we use them for making libraries. And you'll see how we make an antibody library using oligopools in a second. And Biopharma rests on, again, the ability to use libraries as well as custom clonal genes to help others discover, optimize, and develop superior drug candidates for their own internal efforts. As I said, Twist Biopharma, we're a division of Twist Bioscience. We really sit at the forefront of the discovery, optimization, and development cycle. We're not a full-fledged drug discovery company. We do have aspirations to be that someday, but right now, please see us as a company you can go to help you discover as well as optimize antibodies. I've talked a lot about in the past how we're a synthetic antibody library company, but actually earlier this year, we actually acquired a mouse immunization company. You might be saying, Aaron, why would Twist acquire a mouse immunization company when you have such a massive array of synthetic libraries? Well, in my mind, at Varus, who's the company we acquired, again, it gives us another way to discover antibodies. And because Twist can make DNA and has an amazing make test cycle, as you'll see in a second, the more technologies we put at the top of that funnel, the more successful that will be in the end. In addition to the mouse immunization technology that Varus brings, and you'll hear on Wednesday a talk from Varus, we're also working to bring on board a lot of AI machine learning techniques and technologies as well to allow us to curate our data in those other divisions of discovery, from immunization through synthetic libraries. And we're also working to come up with technologies that allow us to discover in silico antibodies using machine learning approaches as well. And so, as I said, we will continue to bring on board additional discovery technologies to really help fill that funnel, because we believe that the world is powered by DNA and allows us to make hundreds and thousands of antibodies, and really at the end of the day, give our customers as many possible antibodies to a multitude of epitopes. I'm often asked, what sets apart Twist Biopharma from our competitors? And I really love to cite these specific advantages of how we are set apart. The first one is breadth. We have an amazing array of different libraries. I'll show you in a second. We actually have 18 different libraries. And a lot of our competitors might have just a few. And the reason why that is it's really hard to make a synthetic library. It sometimes can take even a year to make a high-quality library. But because we're experts in DNA, we can actually innovate and create libraries on the time scale of a month. Second is quality. Of course, with mouse immunization, we get the quality built into the mouse. But in terms of our synthetic libraries, we're actually using oligo pools and actually making natural human CDRs and building them into our libraries. So even though we're making synthetic libraries, they're very natural and high quality because they're based on human sequence. And then finally, Twist is an amazing place for automation. We've automated the entire process of DNA synthesis. It's over 20 different steps. That's all automated. We've actually brought on board a lot of that same automation into our process of antibody discovery. And that allows us to not only do things faster, but also do lots of pannings, lots of screenings, and really power up that MIG test cycle that I talked about before. So in terms of advantages of the Twist offering for libraries, these are a lot of the things I love to cite. But the one I love to talk about the most is that since we're making oligo pools and we can make oligos of different lengths, it's actually very easy for us to vary CDR length, and in particular heavy chain CDR3. In the past, when you had to use a synthetic oligo or a degenerate oligo, you had to use a degenerate oligo to actually make a synthetic library. You would have to make different degenerate oligos to actually give you your different lengths. But because we can use oligo pools, in one run, we can basically make a whole wide array of different lengths of CDR loops. In addition, because we're making specific sequences, we can tailor the diversity within each of those CDRs really finely, and we know exactly what sequences go into the library. It's not just a random collection of amino acids that we know are going into each position, because we actually print in each individual oligo separately in a pool. And that also allows us to remove things, remove restriction sites, remove issues with potential developability of specific sequences. And so in the end, we can actually get back to you a high-quality combinatorial variant library. So how exactly do we do that? So if you think about an antibody domain, let's say a heavy chain, is a collection of 3 loops or CDR loops. We can basically synthesize an oligo pool that encodes for natural human diversity in each of those different loops. And then we can just piece them together and shuffle them combinatorially in the context of a single human germline framework. As I said before, in addition to, again, using natural human diversity as we create those libraries, we can also take away things as we build the library. So for example, we can take away natural human sequences that might have dipeptides that might lead to developability risk. In addition, I won't really get into it much today, we also can insert motifs or insert sequences into our CDR sequences as well. And that's actually key and central to a lot of our difficult to drug target libraries that are focused on G protein coupled receptors, ion channels, and carbohydrates. The core technologies for Twist Biopharma in terms of the synthetic library offering is really, now we have a multitude of different fully human synthetic libraries. We actually have 18 different libraries shown on the left of your, on the left of the slide. We have a whole wide range of Fab, single chain, and VHH single domain libraries. Many of them are fully human naive libraries that can be used for any target. And as I said, we also have libraries focused on difficult to drug targets from GPCRs to ion channels to carbohydrates. On the optimization side, we actually have created this amazing software where we can actually input any lead that you bring to us, and we can actually find natural human CDRs that are similar with a defined mutational distance that we set. And so once we get that information, we can actually make oligo pools and make a combinatorial library where we optimize your lead for greater potency. If your antibody is a mouse antibody, we also can humanize it by putting it into a human framework as we do that to actually give you it's basically a fully human antibody library, a fully human antibody in the end. So the vision for Twist Biopharma, again, is to continue to increase the breadth of our libraries by adding to our library of libraries. It's my vision someday to actually have 100 of them. I think this year we'll actually reach into the 20s, so really exciting, and someday we'll have, and by increasing the breadth of our libraries, we can, again, be more successful at finding antibodies to a multitude of different epitopes on our targets. In terms of our process, we again can do panning on any kind of target from cells to proteins to peptides. We typically do about 3 to 5 rounds of panning and phage display. And then we also then do clonal ELISAs if it's a recombinant protein or peptide target, and also NGS analysis to look to see how our sequences are enriching through the process of panning. Going back to the machine learning approaches I was talking about before, we've been using a lot of machine learning technologies to really allow us to kind of pull out those diamonds in the rough that we probably would have missed through standard clonal screening. So the combination of NGS analysis is coupled with clonal screening has really been impactful and very powerful for us to get all of our hits from our panning. We then feed that into that amazing make test cycle I talked about before, which again, we can make hundreds, maybe even 1,000 different sequences. We port that into our high throughput IgG product that I'll talk about in a second to make purified antibodies in the final format, and then we test them for affinity as well as function. Just to kick off that one product I just mentioned really quickly, actually this week we just launched our high throughput IgG product. It's this ability for not only can you actually now go and synthesize your genes encoding for antibodies, you can actually port that over to a new team within Twist where they'll actually do either a 1 mil or 8 mil transient transfection of HEC293 and actually deliver back to you protein A purified antibodies. So I see that this again as an amazing offering that Twist now has. You no longer have to just order the DNA and do all the work yourself. You can actually get Twist to actually make the final antibody product for you. Okay, so normally at this point in my presentations, I would go through every single library and show you some nice validation data, but I'm not going to do that today. I'm actually going to show you 2 case studies where we've actually used this technology I just talked about for 2 different targets to really show you the power of the platform. The first target I'll talk about is ADORA2A. ADORA2A is a very well validated GPCR target. It's actually been validated by a lot of small molecules that people have found against this target that are doing very well in the clinic. What we set out to do is actually find an antibody that binds and functions better than many of these small molecule comparators. And actually, to date, before we actually undertook this effort, there were no functional antibodies against ADORA2A. So we knew we had a pretty significant feat to go after. And so again, ADORA2A is actually on the end of the adenosine pathway cycle in tumors. The production of adenosine in tumors provides an immunosuppressive environment that actually binds the ADORA2A receptor on T cells and actually puts them in an immunosuppressive state. So if we can block that interaction between adenosine and ADORA2A, potentially can relieve that immunosuppressive environment. Certainly there's lots of other targets upstream of this, for example, CD39 and CD73, and a lot of companies have actually come up with inhibitors of those particular targets and have also shown significant advantage in this space. When we initially went after ADORA2A, we actually put them against the library of libraries. And so I'm not going to tell you that we're successful in every single case, because in that case, we actually did find binders to ADORA2A from our libraries, but none of them were functional. So we knew we had to get a little creative in terms of how we actually discovered antibodies to ADORA2A. And the way we came about it is actually a kind of a merger of both immunization approaches as well as synthetic approaches. And what we did is we actually immunized mice from 2 different technologies. One, the Varus technology with their hyperimmune mouse, as well as a humanized mouse, the alloy mouse. And we actually, once we got tighter in both those mouse systems, we isolated the spleen and thymus and actually did NGS sequencing against both repertoires. We then took the heavy and light chain genes and did a full analysis of the sequence data and actually created synthetic libraries derived from those diversities. And then we then panned and screened them against the original ADORA2A overexpressed on CHO cells. We also made the corresponding immune libraries to those, from those immunizations as well. We actually did get some functional hits from the immune library approaches as well as from the synthetic approaches. This is just showing you one of those antibodies that came out of that endeavor. This antibody TB206-001 is a high affinity binder to ADORA2A shown here, which is comparable to control. It's a very potent antagonist of the receptor and also has very fine specificity for ADORA2A. It doesn't have any cross-reactivity into the other adenosine receptor family members. And also in this case of this particular lead, it actually has some mouse cross-reactivity as well. We then finally did an NVivo study and compared it to standard of care anti-PD-1 therapy. This is in a humanized colo-205 colorectal tumor model. And we actually find that our antibody actually performed quite similarly to anti-PD-1 therapy in this model. Wasn't quite as good as the small molecule comparator, and that's why we're actually optimizing this lead for greater potency, and we believe that we'll be able to get to that same level as the small molecule comparator. Okay, now I'll actually shift over to the next case study, which is against COVID. So in this case, at the beginning of the pandemic, we actually panned and screened our VHH single domain libraries against SARS-CoV-2 S1 spike protein. And we actually use these two libraries, which we call VHH-8 shuffle and VHH-8 shuffle hyperimmune. They're both based on a humanized scaffold. They both have llama CDRs 1s and 2s that we shuffle within this framework. And then in the 8 shuffle library, on the number 1 library, that actually has llama CDR3s in the CDR3 position, whereas the 8 shuffle HI library, we're actually shuffling natural human heavy chain CDR3s. And so from both these libraries, we panned and screened them against the spike protein, came up with hundreds of hits that bound at either single-digit nanomolar, maybe in sub-nanomolar against the spike protein. We went on later to show through both pseudovirus as well as live virus studies against the original Wuhan strain that we had great neutralization in both types of assays. And our top lead was actually TB202-3. And so up until the point of before Delta came around, we actually thought we had a broadly neutralizing VHH-FC molecule that hit all of the different variants of concern. But when Delta showed up, it had this very unique L452R mutation, which is right on the side of the spike. And we had actually submitted our antibodies to the COVID consortium down in San Diego, and they had done a very thorough analysis doing cryo-EM structures. We knew exactly where it bound, which is in this kind of valley on the side of the spike, which doesn't really mutate that much throughout all the variants of concern, whereas the ones that actually hit at the top where ACE2 binds mutate a lot, and that's why you've seen a lot of them kind of fall off with a lot of the mutations. So again, Delta surprised us, and so we actually completely lost all activity of our lead candidate against Delta. Shown down here in the bottom is just some pseudovirus data. These two lines down here are actually both variants of concern that have this L452R mutation. So at this point, we felt quite defeated. We had basically gotten a good lead, and then we actually lost almost all of our activity with this particular mutation. So we didn't give up. We actually then went forward and actually went back to the original panning that we had done, did some additional rounds of panning on some other variants of concern, including those that had this mutation, and actually pulled out an additional VHH candidate called TB339031. We did an alanine mutagenesis study with integral molecular, and this new antibody and TB203 actually have a very closely tied epitope, but they actually have some differences in their overall epitope. And the cool thing about TB339-031 is it actually does now bind Delta and actually had very good neutralization against both pseudovirus as well as live virus with the L452R mutation. As we were doing this, Omicron showed up. So we actually found when Omicron reared its ugly head that the original TB202-3 lead actually did great against Omicron, and TB339031 actually didn't. So because we had these two VHHs that had really distinctly different activities, we actually put them together in a bispecific and actually showed that the final molecule binds both Delta and Omicron really well as on the right, actually neutralizes both viruses. Of course, when we go against a virus where both of them bind, like the original Wuhan strain, we actually see an even more avid interaction and a better neutralization because we actually have four binding sites on the bispecific rather than just two. All right, since I have a little bit of extra time, I'm now going to talk about a new library product offering that Twist is rolling out, and it's really to help our cell therapy colleagues that are trying to come up with either engineered TCRs or engineered CAR-Ts for their particular cell therapy. And on the engineered TCR front, there's a lot of companies that are, again, isolating patient-derived samples and trying to pull out the specific T-cell receptors on these TCRs that are actually having an anti-tumor effect. And so, because again, Twist can actually synthesize a lot of DNAs, we're actually working closely with many of these partners to come up with libraries where they can actually shuffle all the various TCR alphas and betas together to really pull out and tease out those specific alpha-beta combinations that have potent anti-tumor effect. And the way we do that is through a new early access product that we call a combinatorial assembly library. So here we can actually make up to 100 TCR alphas as gene fragments and up to 100 TCR betas as gene fragments. And then we can basically shuffle them together. So if you're doing NGS analysis of your TCR repertoires, you can basically take the top 100 of both, link them together, and we can actually make a combinatorial library where we're shuffling them into a 10 to the 4th library. We have 100 TCR alphas and 100 TCR betas to create a 10 to the 4th combinatorial assembly library. So this is an amazing way to, again, survey the combinations of alphas and betas in your data to give you potentially a number of different potent T-cell receptors that might be giving you that anti-tumor effect. And so we believe this is really game-changing for the space, allows people to not just use specific TCR alphas that come out of their specific work, but also look at the combinations of different T-cell receptors to see if they can pull out even more avid interactions, more functional interactions. And again, all of these libraries, like all of our libraries that we build at Twist, is very well validated for even distribution of each of the members in the combinatorial library that we build. We can also use the same approach with CAR Ts. So again, if you think about a CAR T as, again, a targeting domain at the top, a hinge region, a transmembrane domain, and then a series of signaling regions, we can basically make gene fragments for each of those different parts of a CAR T and then just shuffle them all together. You may think, well, most people, they just use the same hinge signaling domains in every CAR T they do. But what we're finding and working with a lot of our partners is that depending on which targeting domain you use, those other elements may actually be better optimized with other specific units. So we're working with others to actually come up with a library of CAR Ts, not only having a number of different target domains, but again, a whole wide range of different hinge, transmembrane, and also signaling domains to allow you to optimize your CAR T for optimal display as well as optimal activity. To come up with your target domains, of course, you can use the libraries I talked about before for helping you discover both either an scFv or a VHH that goes after your target. Or you can also work with our combinatorial variant library team to actually build your own library that you can, again, pull out your specific binder to serve as your targeting domain. But then after you have that targeting domain, as I said, we can create a combinatorial assembly library where we put in representative members in each of those modules that we synthesize as non-clonal gene fragments, and then we can just shuffle them again on a library up to 10 to the fourth. And we actually did a really elegant study with Serotyny, which is another really fantastic SynBio life science company, where they actually, we worked with them to create a CAR T library using known targeting domains that were already known against a number of very well-validated CAR T targets. And they had a really nice screening system for looking at, within the library, which CAR Ts were the most functional. And so they actually did a whole series of assays looking at activation, proliferation, and cell killing within the library that we built for them. And they were able to pull out a whole wide array of optimal candidates, each specific for each particular target domain that they actually put into the library. So just to summarize again for CAR T, we can make a library up to 10 to the fourth built off of non-clonal gene fragments. This allows you to really look at the space of possibilities, not just for the target domain, but also for the other various elements of a CAR T to give you the optimal presentation, activity, proliferation, and cell killing within your particular system. So just to conclude, we're here to help. We have a number of technologies that I kind of rushed through today, but I hope you see that everything that I've shown you today is powered by DNA, from the amazing make test cycle that we can make antibodies and do all the downstream testing, all the libraries that we can create, all the work we can do with other divisions of the company to bring in other leads that we can then put into the make test cycle. And so again, if you have any needs in this area, please come and see us in our booth. Thank you for your time and attention. I'd be happy to answer any questions.
Hello, great talk. Quick question. I noticed in your VHH library that you had about, I think, 1,600 CDR3s. Why not use more? Is there like a reason? I mean, you have a chip that can synthesize 1 million sequences, so why not have 1 million CDR3s?
Yeah. So for those series of libraries, we actually started with a series of 3,000 VHHs it was within a specific set of llama sequences. So that's the set we started with, which was a little small. So that's why there was on the order of thousands in that particular loop. We've now since gone back and we're actually working on a new library based on camel diversity, where we're actually really powering it up. I mean, all the loops. So stay tuned. There's going to be another library we're going to put in tens and hundreds of thousands of sequences in CDR3 in particular.
Cool. Thank you.
Jim.
Hey, Aaron, really great talk.
Cool.
I wondered, in terms of the numbers of libraries that you think are needed, when do you know you've made enough? You've got 18 now. how do you recognize there's holes in this, in the sort of the search algorithms that we need to fill? What is your thinking about that? And can you, combining these, is that better than just making a super large diversity library?
Yeah, I mean, in my mind, the space of possibilities for sequences is almost infinite. So we're never going to be able to cover all space with the set of libraries that we have. That's why I think we should continue to come up with more and more libraries as part of our libraries. But with that said, I think there probably will be a stopping point. I like the idea of being as creative as we possibly can to use different frameworks, different diversity sets to potentially isolate different pockets of that allowed diversity. But I agree with you. I think at some point we'll have enough overall diversity and number of scaffolds as well as different frameworks to really cover our bases within the space of allowed diversity. All right, thank you so much. Please go check out our booth if you want any more information about Twist Biopharma.
Thanks, Aaron, for a great talk, and I invite everyone to get a cup of tea or coffee, and then don't go far. We'll start here at 4 p.m. for the plenary keynote session. Thank you.